Good afternoon to all of you in the room, and good day, whatever time of day it is for all of you joining us online. Thank you for joining us. So welcome to the fourth Scientific Colloquium. This one is on establishing R&D consortia in Southeast Asia to advance mRNA vaccine development up to a proof of concept. And so first of all, I'd like to welcome Martin Frieder from the World Health Organization to give some introductory remarks. Martin.
Thank you, Charles. Once again, quick sound check. Can you hear me?
Perfectly.
Perfect. Thank you. So welcome, everybody. And once again, my apologies that I can't be with you today. I was really looking forward to this meeting. So a quick reminder of how we've come to this point. During the COVID pandemic, it was very clear that the low and middle income countries were the last in line to receive pandemic vaccines. This is not the first time this has happened. We saw this during the influenza pandemic in 2009. And we also have seen on multiple occasions that vaccines have in the past really been developed by the high income countries. So WHO, along with its partner Medicines Patent Pool, set out in the middle of the COVID pandemic to transfer mRNA technology to...
Martin, we lost you. I don't know whether you clicked onto mute by mistake.
No, sorry, it could be the internet in Geneva is quite bad today. Charles, can you hear me? Okay,
fine now, yes.
So just where we've come from, WHO and MPP set out to build mRNA production technology in all the regions of the world. But we know that if we only transfer the technology to make COVID vaccines, this will not help in readiness for the next pandemic, because if you can only make COVID vaccines, your factory will soon go out of business. There is decreasing demand for COVID vaccines. So to support sustainability, we are now actively trying to promote research and development of commercially viable products that will contribute to health in low and middle income countries. But the development of these commercially viable products, this is generally not going to be feasible for the manufacturers whose main focus is manufacturing, not research and development. Therefore, to support this, we have tried to pull together research and development, what we call translational research units, especially beginning off in Southeast Asia, and to identify what products could be made and are there groups with the adequate technical know-how of how to do the research and development of these. So we have 4 potential targets that we began discussing first in South Africa last year, then in Bangkok, and now today. And these are the hand, foot, and mouth disease, Enteros 71A. It is dengue. It is human papillomavirus, but not a prophylactic, but rather a therapeutic human papillomavirus. And finally, Plasmodium vivax, the malaria. And we hope that several, or hopefully all of these, will become commercially viable products that can be manufactured by the partners. From the WHO and Medicines Patent Pool perspective, what we really are aiming for is that it is open access, that the results of this research and development are shared openly with the members of the WHO MPP mRNA Manufacturing Consortium. So really, just to summarize, I remind everybody that the objective here is health, first of all. It is commercially viable products for which there is going to be sustainability. And this is going to support future pandemic preparedness in all parts of the world. With that, Charles, I hand back to you. Thanks.
Thank you very much indeed, Martin. So we've got 4 presentations on the various consortia for the 4 diseases that Martin mentioned. And so first of all, I'd like to invite Jerome Kim, who's the Director General of the IVI in Korea, the International Vaccine Institute, to present on dengue.
Thank you, Charles. Okay. So starting off with the challenges, and I'm summarizing from this morning and a bit from some of the review papers. 3 vaccines, 2 of them WHO qualified, a third on the way, but as Q pointed out, 1 is not usable, 1 has partial coverage, and 1 has unknown coverage of dengue 3 and dengue 4 types. Another extract from this morning, there are 4 dengue types, each capable of causing infections, disease, and death, each with differences that may be distinctive and quantitative. And that idea that all of this, that we should think of them as different, I think was a very important one. The absence of validated immune correlates of protection was discussed extensively. Animal models, the fact that animal models don't really fully mimic human dengue infection or disease, again, was brought up. And when you talk to someone from HIV, the idea of doing study after study with vaccines and the wrong virus or the wrong animal models is really something that we do pay attention to. The fact that immunological assays are unable to precisely define dengue type specific immune responses, the requirement for very large efficacy trials to demonstrate benefit across diverse populations and clinical endpoints, the possibility that CHIM may be able to make up help us to solve some of the issues, particularly around correlates of protection, the potential need, which was just hinted at this morning, for large effectiveness trials. And could this be a substitute somehow for efficacy in certain circumstances? Again, I think it was raised again indirectly this morning, but something that people have talked about before. And then something that we really didn't talk too much about, which is around, we're going to be making these vaccines regionally, but who's going to procure them? So is there a procurement mechanism like AVMA that has been proposed for Africa, or implementation? And as we get to these, we're getting to parts that don't correspond to manufacturing specifically, but have a really significant impact on the manufacturer. So, and again, I don't think these are the right slides because there should be a warning here that this has not been fully discussed with our collaborators. Actually, it hasn't in some extent been discussed at all with some of the people who are listed on the slide. So apologize if your name is on the slide and you haven't spoken to us, because this was developed specifically for this meeting and in a bit of a hurry. So a high-level budget and some basic timelines for 5-year, 4 years to the start of phase 2, and through the initial proof of concept in humans. So again, very high level, very non-specific. As we get into more of the details, things will undoubtedly change. Hopefully they won't slow down. So assumptions considered for the initial plan: preclinical and process development will be done in parallel with manufacturing partner selection processes. They will produce batches for both toxicology and phase 1. Toxin stability results will be submitted to the NRA of the country where the clinical trials are going to be conducted once available. Preparation of first in human will proceed in parallel with batch production toxin stability studies. And the phase 1 clinical trial will be about 80 adults with 2 doses, hopefully. This is, again, a high-level Gantt chart. I think we were asked to provide this. And you can see it roughly corresponds to the timeline that I'd mentioned before. You can see where the batches are produced and where we start to get into the first in human studies in year 3, and then the phase 2 starting at the end of year 4. A partial list of stakeholders. So you can see preclinical and process development at Chula. Technology transfer, if IVI or Hillman need to support this, then this is something that we've done for others. Global access, again, typically we put the global access agreements in a subaward terms, and we often use the CEPI or Gates versions of global access, which usually correspond very broadly to a manufacturer committing to provide the vaccine at very low cost for global health purposes. Manufacturers, we know Biopharm and Incepta are here. Other manufacturers are possible because there is manufacturing already available through time manufacturers, for instance, and contract manufacturing operations and potentially scaled up. Toxicology studies will be conducted as required by the regulatory authorities of the country of manufacture and as needed for WHO PQ. Companies will do the regulatory work, although IVI can actually support or even function as the sponsor under certain circumstances. Clinical trial conduct, there are potentially lots of different options, and this really needs to be discussed because it will depend on the funder and where and what the vaccine trials are being conducted for. That could change the scope of where the trials are actually conducted. Will they be conducted in one country, multiple countries, and the efficacy trial, will it be conducted across the region or primarily for the purposes of a single country. Again, it'll depend on the regulatory strategy that's agreed to with the regulatory authorities. Clinical immunology assays. And again, IVI has done qualified and validated assays in support of clinical trials. Other organizations can do them as well. We are happy to develop and technology transfer these assays, as we have for multiple assays in the past, including during COVID, transfer of validated clinical assays in support of clinical trials. Very important, but also very important technology for other parts, other countries and organizations to be able to do. Consortium and partner management, and IVI has done this for multiple studies and vaccines in the past. So next steps. Oh, there's the caveat. These are notional, have not been discussed with partners, and are subject to change. So in terms of immediate next steps, a memorandum of understanding, exploration of funding opportunities with preparation of a consortium proposal. I didn't mention it on the overall budget, but we're thinking between $7 and $8 million to take us through phase 1. Consortium agreement in place with partners defining roles and responsibilities, scopes of work, timelines and budgets. Clinical development plan and/or integrated product development plan, depending on if you're Gates funded or not. You know what an IPDP is, integrated product development plan, but it really covers the clinical development of the vaccine in question. And the IPDP often goes beyond just the clinical trial itself, but into the other things that we need to think about in parallel. Agreement with manufacturing partners, technology transfers, know-how, intellectual property and timelines, a regulatory strategy, a pathway, and identifying a regulatory authority that will be our principal correspondent during this. And this is really important because, as we look outside of an individual country or at WHO pre-qualification, the regulatory strategy becomes very important for us. Establishing a joint steering committee and technical advisory committee. So the difference here is the Joint Steering Committee has a responsibility for the conduct of the collaboration and the structure of the dengue vaccine consortium. The Technical Advisory Committee is usually composed of experts, people who are expert in dengue and/or clinical trials or regulatory, and they provide very concise and necessary advice to the people conducting the trial. Often, we prefer to have people who are outside of the organizations that are principally involved. Again, because sometimes we get this kind of unique tunnel vision and we don't see the things that we should be thinking about or seeing. So this will be the governance structure for the consortium. The steering committee will review the progress and collaborative activities. Of course, things that we do in other organizations like the Hilleman are all project management milestone driven. So they're driven along this pathway that is determined in advance of the actual start of the trial, start of the effort. And the steering committee will also be facilitating communications and decision-making processes between the partners. So I think anyone who's been on a large collaboration understands that this is something that is complicated, requires lots of communication and openness between the partners. So we can't have a group doing something that they're not informing other members of the consortium. At the same time, and again, this came up this morning, and I can't overemphasize that we don't have the answers on the science yet. So as we're doing all of this, the science is going to progress. Qualification and validation of assays, work on correlates of protection, or at least, for instance, and this came up this morning, a cellular composite, maybe a correlate of protection that is cellular, that is based on multiple different cellular assays that we can put together into a testable hypothesis. Cost effectiveness, and updating national and regional key opinion leaders and stakeholders. Because again, the last thing you want is to be done and then have to talk to people and then hear from one person or a country, oh, you can't do this. You should have been thinking about doing this first, or you need to do an effectiveness trial. And we didn't have the funding or the planning around how we were going to do that. Because that just means then we don't have money and then we have to struggle for funding to secure the money in order to do effectiveness trials. So this is a part of the total plan, and you have to think with the end in mind, and I'm at 22 seconds left, so I'm done. Thank you.
Thank you so much, Jerome. And next, I'd like to invite Raman Rao, the Chief Executive Officer of Hilleman Labs here in Singapore, to present on hand, foot, and mouth disease. Raman.
Thank you, Charles. Good Good evening, good morning, good afternoon, everyone. Jerome highlighted in depth some of the challenges and potentially the pathway towards a multivalent dengue vaccine. While the team tomorrow, the consortium team, will discuss more in depth, I just wanted to provide more highlights on how do we envisage potentially a similar approach for HFMD. Just to highlight, HFMD shouldn't be confused with FMD. FMD, foot-mouth disease, is a veterinary disease caused by virus belonging to the same family, but this is typically predominantly a pediatric disease, and it's very prevalent, and it causes a lot of disease. Mostly it's sometimes very unknown because it doesn't cause significant mortality. Although we've seen the evolution of the disease wherein it's been where we've seen more and more cases potentially of mortality, especially neurological illness. It's again very common in children. It's a pediatric disease, 2 to 5 years of age, caused by the host of viruses belonging to the enterovirus family. The enteroviruses, the Coxsackie viruses, and it's the same family that polio belongs to as well. So it's a Picornaviridae family. Most of the transmission is fecal-oral, so it's very easy to transmit. It's extremely contagious. And there's a lot of shedding of the virus, as is seen with traditional enteroviruses. Again, as I had mentioned, the virus is very, sometimes a lot subclinical. Overt clinical symptoms are generally fever, soreness of throat, and then you have blisters. But what we've seen is, as the complexity of the molecular epidemiology of the virus has changed, we're also seeing a little bit of more case fatality. And that's what has evolved over the past 2 decades in the region. And when I talk about the region, it's primarily in Asia. So when we look at the disease per se, the hospitalization rates have increased, especially again, that close to 6 to 10%. And amongst those subjects who are hospitalized, you see some of them moving towards CNS complications. And that's where we see the case fatality rates going up. And mostly, as we've seen with EVA71, enterovirus A71, that evolution is much wider than what we see with Coxsackie viruses. So if we see across the continent of Asia, as I'd mentioned, the virus belongs to the family Picornaviridae, very similar in evolution to the polioviruses, but very different structurally. And that's where we see some of the capsid structures being very important as we look at the vaccine design. And we've seen that with existing commercial vaccines as well. But what we also have seen is across different countries, whether you look at China, Japan, Korea, regions of Hong Kong, Singapore, Vietnam, we've seen the virus's epidemiology change as well from enterovirus A71 through to the Coxsackie repertoire of either Coxsackie 6, 10, 16, 18, and although that has been progressively how the epidemiology has evolved across the region. And what that entailed was a significant burden. And when we say significant burden, it's more in DALYs. And this is within the pediatric population. So you have school closures, parents having to take leave from office, and all of that adds up. And the economic value proposition for vaccination has significantly increased because we see the changes or impacts in terms of DALYs across the continent, across different countries, including Singapore as well. Locally, we've had a lot of outbreaks. What we've also seen is a change in some of the landscape in countries like China. And the reason being, China is the only country which has 3 different commercially available vaccines, and they have implemented a program of vaccination in the pediatric population. And what that has created was a reduction in the case fatality, but the case burden has remained fairly significant around the same number of 2 to 3 million cases in the year. Whereas if you compare that with a country like Malaysia, where you see the cases going up and down, but it has been a progressive increase overall in the disease burden. So that's what is really the scenario when we look at enterovirus and Coxsackie virus disease or hand-foot-mouth disease in general. As I'd mentioned, it's one of those diseases where we have 3 commercially available vaccines, and these are based on the traditional inactivated whole virion vaccine approach. And it has been fairly significantly successful in China. There are 3 vaccines that have been commercialized with very high efficacy rates. And the challenge has been, how do you then evolve a monovalent approach to a multivalent approach as the epidemiology has been changing? And that has been the challenge that has been facing manufacturers and also vaccine developers. So apart from those 3 successful vaccination strategies, we have not seen the evolution into multivalent targets. And that has been the lacunae in the field over the past decade or so, where we've seen the disease evolve, but the vaccination strategies haven't evolved as much. So consequently, as I'd mentioned previously, in China, we've seen the case rates being similar, whereas the case fatality has come down. That is primarily because of vaccine preventable diseases. And that's what we see between the years until to the end of 2015, where there was a significant public health strategy for vaccination. The case loads had come down. But what transpired after that was that the cases went up because of the other strain replacement happening with Coxsackie viruses. So that's a gap that has been identified and which needs to be addressed. And what we hope is, while we have some commercially available vaccines, they're still monovalent. We need to have multivalent strategies. And that's where we believe potentially an opportunity for mRNA vaccination, mRNA vaccine approaches, because of their obvious advantages. Of course, we do understand the challenges with mRNA. We've not used mRNA for a complex virus family like picornaviruses, but because of the capsid structure and structural implications of the way the viruses are, there is a feasibility that we can potentially look at multivalent approaches using mRNA. That can then help in translating most of these technologies across different partners in the region who can then implement vaccine strategies. While all of this is happening, we do also understand the challenges of multivalent approaches. I mean, we discussed that with dengue. It's a similar scenario, albeit being a same family of viruses. We do think that you will encounter those challenges, but hopefully with mRNA approaches, we'll be able to surmount those challenges. So that's where the consortium comes into the picture. And the goal of the consortium is to get into clinical phase 1 studies with a proof of concept for a multivalent approach. And hopefully we'll have EVH71 and some of the Coxsackie viruses we talked about. Because of some of the challenges with these viruses structurally, we hope that the consortium approach will be able to create the right constructs, and then we'll be able to translate that into product development strategies with CMC expertise and get that into clinical phase 1, following which the partners in the region, Vietnam, Thailand, and Indonesia, would then be able to carry forward until commercialization. And the consortium approach, as we mentioned, is extremely important, and we're trying to bring in together a significant ecosystem of partners who have a lot of value proposition within Singapore and outside as well. And we'll be working closely with these partners. You'll see the entire plan with the team tomorrow, but the idea is that partners with NUS and A-STAR would bring in the early development, construct development strategies and the preclinical development, because it's important to emphasize that the preclinical development involves a lot of challenge experiments. And although they are small animal experiments, there's a lot of challenge experiments that need to be done both as monovalent approaches and then multivalent put together. That will then be translated by Hillerman and the team, along with working with the BTI Bioprocess Institute, to create the initial preclinical lots and ultimately into phase 1 clinical studies. And that will be used ultimately to tech transfer into the commercial partners. So we hope that working in tandem with the consortium partners, and the entire plan is something that the team and the consortium partners will discuss tomorrow and bring forth for a successful development of a multivalent HFMD, hand, foot, and mouth disease vaccine. Thank you.
Thank you very much indeed, Raman. And now I'd like to ask Kiat Ruxungtam to come and talk about therapeutic vaccine for human papillomavirus. Over to you, Kiat.
Yeah, thank you very much. So this HPV therapeutic vaccine development consortium is actually we start a few months ago, so the part I'm going to present to you just to show that it's feasible that the R&D can move fast and hopefully the consortium can be more identified and how to move forward. And we're going to share with you some of very promising results. So the July Vaccine Research Center, our commitment is for more vaccine access to low and middle-income countries. And the Vaccine Research Center has been founded a decade earlier, but the official name just 2 years before the COVID. And together with Drew Weissman, we have a clear commitment that we would like to bring this technology platform to make it more accessible. So the capacity in terms of R&D, research-grade vaccine in our facility during the pandemic, so 4 years, we have capable to cover most of the value development chain of the R&D grade candidate vaccine. So right now, that's why we are very honored to have a chance to join this Southeast Asia consortia. So in terms of HPV, we all know that HPV vaccine preventive is very effective. But up to now, again, this is based on the WHO, some of the data, 2020, only 1/5 of people or 1/4 of people accessible to this vaccine. And when we look at the people that are already infected with HPV, women globally about up to 300 million people infected. Male has been a number of prevalent studies show that about 1/5 of male also had been infected. And the death rate of the cervical cancer related in women has been estimated by one of the WHO draft report on the HPV therapeutic vaccine, TPP, is about every 2 minutes, one woman die from cervical cancer. So the rationale to develop HPV therapeutic vaccine, and this is the first draft of the WHO preferred product profile, to increase global public health value, at least should cover 2 major oncogenics, high-risk oncogenics types, is 16 and 18. The goal of therapeutic is, the first goal is to reverse the pre-cancer state of the disease. So from SIN1 to SIN3, and regress to either SIN1 or normal. And/or to clearance the HPV DNA, or prolongs the effect against reinfection or recurrence. Now, I'd like to share with you some of our very promising results. And again, this development is started when we have the first meeting in Bangkok with the team of starting the consortium. So the first thing is that we develop. So this is my colleague that the key driver of the programs. So we investigate 3 antigens, 1, 2, 3, and all the protein expression is very impressive. And the first thing to start this type development is to set up the tumor mouse models. And Supicha have developed this from scratch with the collaboration with a collaborator in the US who provides T-cell 1 luciferase cells. And so the first question is, what is the model that addressing early tumor development? And that will be used that. So we explore the optimization of the implantation of 5,000 cells, 10,000, 20,000, and 50,000. You can see that the tumor load in the implantation model is really, the progression of the tumor is faster in the higher tumor cell load. So 50,000, you start to see, sorry, I don't have a pointer here, but you can see that with the 50,000, you start to see the tumor growth, the tumor size by naked eye by day 3. And with a 10,000 cell, it's going to start to see clearly about day 7. And this is the fluorescence imaging that you can see. Now, the first question is, actually, we have done quite a number of studies. The first proof of concept is whether 3 doses of mRNA can make a significant regression or tumor-free in the animal model. And then the answer is yes. If we go with a 50,000 cell, it's reduced more than 10-fold of the tumor size, but you won't see a tumor-free. In this model, so we asked the question that, and the two doses of mRNA vaccine also see 100% of tumor-free results. Now, the question is, can only a single dose of mRNA vaccine, this is using HPV-16 as a model, elicit tumor cell regression in mice after giving 5,000 or 10,000 tumor cell implantation? The answer is yes, and you can see that it's very clear that the normal cell in control, you see the tumor growth, and the vaccine antigens, you have seen clearly that either the 5,000 cell tumor cell load or 10,000 is completely shut off the tumor growth. And again, this is not the L1 antigen. So we also design as experiment whether this can do 2 jobs. So I mean, if you immunize before implanting the tumor cell, can that protect the animal in this model as well? So we gave the 2 doses of HPV mRNA vaccine candidate, and then implant the tumor cell at the number of 10,000 cells. The experiment, again, this is unexpected. It's also completely, induce the tumor regression. So again, in summary, it's a very early, very young consortium, but we're seeking more partner in the consortium. And of course, we have to deep down further about the scientific research question and gap, and how to further design the antigens and which antigen. To us right now, it's also only single component of the antigen is enough to generate the tumor-free results. So the next step is to expand the consortium, and then to see how we're going to move forward. Thank you very much for your kind attention.
Thank you very much indeed, Kiat. And our final presentation is from Jetsaman Setabongkot, who's the director of the Myodol Vivax Research Unit in Thailand, on the Consortium for Plasmodium Vivax Malaria. Jetsaman.
Thank you very much. It's my honor to join the colloquium. So among all the consortium, I think I'm the youngest. We just established 2 hours ago. But anyway, we initiate a collaboration with Professor Kiat Group for the past, I think, more than 6 months. And what I like to show to all of you is that, especially for malaria, no one that can work on malaria alone. And in fact, many consortia established for other type of malaria. This is the first Plasmodium Vivax mRNA vaccine consortia that suggested by the WHO in the last October in Bangkok that we should have it. And I totally agree, because working together, you can move things fast. And with this platform, that's supposed to be the fastest way to produce the candidate vaccine to go to the trial. So I was very excited at that time last year to meet the other collaborators or potential new consortium today that we can join and complement each other to move this field forward. So I'd like to spend a few minutes only to summarize, just to show you how we had involved with the mRNA vaccine for Plasmodium vivax. You know, as a researcher in malaria over 35 years and working related to the vaccine and drug, the problem of the vaccine in general for malaria is that any in vitro system in the lab always work more than 100%, fail 70% in the bigger animal up to the monkey, and completely or almost completely fail many times in human. The time between you try to produce the protein to be used for the GMP lot and for human is always take long time because the parasite protein is so complicated. And if you need among all, I'm the only woman and I'm the only one that work on parasite. All the gentlemen, they work on virus. So this is the most complicated. When we learn about mRNA vaccine platform, that even before the COVID, but I think we benefit from the COVID and the vaccine for the COVID that happened to be mRNA. So everyone's so familiar with what you should expect from that, and also for us. So the first part of the vaccine that we are looking for is called transmission blocking vaccine. The reason that we choose to try this as a candidate to compare if it's more superior than the protein platform, because we had experience to work with this antigen for a long time, since the mouse model until the human model trial many years ago with the collaborators at NIH and at other institutes. So we tried this, and then from the funding support, the first phase by the GHIT funders from Japan, we, in collaboration with UPenn, Dr. Norbert Pradi, used the platform of the modified mRNA to produce the first candidate and prove that it's functioned very well. You know, it's superior than the protein vaccine, as you can see in the figure, to show that the duration of the antibody response stay longer and higher. We now have the project ongoing to work on the liver stage of Plasmodium vivax, and it's ongoing. But to move the project further, we met Professor Giet and then discuss about how we can do it faster within Thailand, right? So Chulalongkorn and Mahidol University is not that far. So we met at the other meeting and then discuss. So because Professor Giet get the license from UPenn to do the platform, exactly what we did before, and then but just use different LNP. But anyway, we go for that and try the first candidate, PVS25. It look very good. And further from discuss with him, we also learn a lot more how to move forward to the non-human primate and start working with Professor Sujinda, which already presented the talk in the consortium discussion. So all of these happened within less than 6 months. Now we are ready to go for the non-human primate. And if we get the first result from the monkey study that promising, we're ready to move forward to the human trial, the phase 1 trial. Because our group already established the CHIM, or control human malaria infection, at the faculty that can test all stages of PVivac vaccine. And partially already got fund to do further phase 1 trial in human. If we can get any candidate put in the pipeline before the end of 2026. So a further discussion has been made with the pharma, okay, biopharma, if we want to go to the human, who can be the potential industrial partner. So we had a virtual call a little bit to brief to prepare for this meeting. And this is the timeline that after discussion, this is a potential timeline for all stages of vaccine. The blue one is the vaccine stage that we already work. The yellow one is the blood stage mRNA that the partner in the group are interested. Dr. Rintis from Indonesia has a lot of data on the blood stage vaccine for other protein, but not mRNA vaccine. So I think combine our expertise and resource in each group that we have. For example, Indonesia and Thailand has a lot of cases. We can do similar study in terms of academic, and we can help set up more human challenge model in Indonesia if infrastructure is ready to go. And then this is a timeline. I don't think is overestimate. I think we can do it. You know, by 2027, if the antigen, if the candidate really look promising, we should be able to have at least a few phase 1 trial in Asia for Vivax mRNA vaccine. And this is the current institute that agreed to join, you know, because they sent me the logos. I assume that everyone confirmed. Okay. You can see that Mahidol University and represent Mahidol University, although we also collaborate with the other group. Okay, Burnett Institute, Dr. Herbert represent, and we hope that we can jump into his group expertise in terms of immunology that can explore better vaccine candidates. Chulalongkorn University, lead by Professor Kiet from Jula VRC and Professor Suchinda from National Primate Research Center of Thailand, which is, I'm glad to meet her because we can do a lot more, not only the mRNA vaccine. And EHI and BRIN from Indonesia and Biopharma. So thank you.
Thank you very much indeed. And this is very exciting. And could we have a round of applause for all of our panel? Thank you. Okay, so now we're going to have a short session where we're going to have invited the 4 participants here to sign the Declaration of Commitment, which I'm going to read out in a second. And anyone who in the room who would also like to sign this, we'll have a period at the end of the session where we'll do a group photograph, and anyone who would like to sign, we would invite you to sign. I shall read it out first. We, the undersigned, understand the immense potential of the mRNA technology transfer program to build outbreak response capacity in delivering important health innovations for and by low and middle income countries. We further understand the importance of ensuring the long-term sustainability of the selected manufacturing partners, so that each partner will stand ready to respond to the next pandemic, to contribute to greater health equity in LMICs in times of public health emergency. Such sustainability will be facilitated by the research and development of mRNA products for routine use between pandemics. We each bring expertise in health, product research and development, and a willingness to share the results of our R&D on a fair and equitable basis with the partners of the mRNA Technology Transfer Program. To that end, and pending the availability of adequate financial resources, we commit to collaborating on research consortia to develop mRNA vaccines targeting the following 4 pathogens: dengue, enterovirus A71 Coxsackie, human papillomavirus, and Plasmodium vivax, with the aim of generating proof of concept preclinical data, the preparation of phase 1 material, and the conduct of the phase 1 study. We further commit to the sharing of the material, data, processes, and any resulting intellectual property on an equitable and non-exclusive basis with the partners of the mRNA Technology Transfer Program, with such commitments to be further defined in subsequent agreements. Thank you very much. Thank you very much indeed to our panel. And yes, I have signed. So with that, I will now invite our second panel to come up for a discussion. So Petro, Hani, Eduardo, Q, and Gabrielle, if you would like to come up. Yes, no. Okay, before I start, I'm just going to introduce our panel. I'll go from left to right. So Q Yun is the Acting Executive Director of Research and Development at CEPI. And Petra Blanche is the Managing Director and Chief Executive Officer of Afrigen Biologics. And Hani Kin is the Executive Director of the Wright Foundation. And Eduardo Banzon is the Principal Health Specialist at the Asian Development Bank. And Gabrielle Leong is the Executive Director of the Hong Kong Jockey Club Charities Trust. So I'm going to start with you, Petro, if that's all right. So in what way do you think that these regional R&D consortia will enhance the ecosystem? And what is your vision of this going forward? And maybe even what do you see as the ecosystem that will need to support the consortia?
Good day to everyone, our online participants also. Charles, thank you very much for the opportunity. First, a comment. For mRNA or the RNAs to be well embedded in contributing as a vaccine platform, a development and manufacturing platform, we need to advance the science. What this consortium is going to do, and has already started to demonstrate, is the ability to advance the science end to end, from the discovery of antigens, the design of antigens, process development, scale-up, preclinical development, clinical development, and ultimately commercial production for public health benefits. The participants and the consortium members here all come as centers of excellence. They have deep science, deep understanding, and they have platforms that they bring to a consortium that is focusing on particular unmet needs in the region. I am very excited to see this consortium developed and to see the progress in a very short period of time. So for mRNA to be sustainable as a platform, we need products. We need products that goes into the public health and contributes to public health and bring manufacturing sustainability for the partners. I have seen in the last day what has come together here, Charles, as public, private, and academic partnerships. It was good to see the partners of the mRNA technology platform, Incepta, Biopharma, and Polyvac present here, participating in these consortia, driving ultimately the sustainability of the program. What is my vision for this? That this will advance and fast-track vaccine development or vaccine candidate development for mRNA at a global level, and that the knowledge sharing will contribute to the sustainability of the technology transfer program and its partners.
Thanks very much indeed, Petro. And I should have said at the beginning, we will have a reasonably short session at the end for Q&A, both for those in the room and those online, if you want to post questions. So next I'll turn to you, Hani. Similar question. What do you see the relationship between this consortia and the ecosystem, and what is your vision for the future?
Thank you. I would like to thank the organizers for inviting Wright Foundation for this important discussion. I was deeply inspired by the level of collaboration that's happening in this region. And for me, I think the biggest significance comes from the potential of these nascent R&D consortia to serve as a model. I think, Charles, you used the word template at the end of the session today, to serve as a model of a platform for regional collaboration to drive R&D to serve public health needs, both regionally and globally. So I believe what we have at hand is a task and an opportunity to prove the concept that regional collaboration can be done with committed partners, with shared objectives, as well as with a focus, which is what we've seen during the earlier presentations today. And not only can it be done, that it can succeed in developing and making available essential health products as public goods to actually have an impact on regional public health. I believe that if that concept were proven, then we'll be able to expand the list of diseases to include other diseases that have unmet needs for improved or new health products, and to include products beyond just vaccines. And that is to say that we will be in a position to build a regional R&D pipeline. And the reason why that is critical, I believe, is it's going to allow us, different actors, to contribute in a complementary way to collectively cover the full value chain of R&D for public health needs in the region. And that's going to be hugely transformative in coordinating collaboration to maximize the impact of limited resources. So I'm hugely excited to see the beginning of that efforts happening in this room today. Thank you.
Thank you very much indeed, Hani. So next I'll turn to you, Eduardo. And again, what do you see as the relationship between these consortia and the ecosystem, and what's your vision? You're on. No? Press it again. Yes, you're on.
Okay. Probably need to press a little harder. No, I always like to reflect on what during the pandemic. Sorry, can you hear me? Where we always try to say that regional collaboration, cooperation with developing countries, ASEAN, was something that hopefully happened when we tried to respond to the pandemic. It was difficult. We're not even talking about how developing countries, the relationship, but even with ASEAN, trying to get, for example, getting the different countries to procure together, having discussions where if there's manufacturing, for example, in biopharma, and they prioritize other countries, it was just not happening. I think this is where the RD consortia is going to change the ecosystem. We in ADB will always talk about the value of regional collaboration and integration. We have done a lot of work in getting countries to work together, from the Greater Mekong, who just came from Malaysia, Bimpiaga. And we always talk about sitting down countries and say, okay, let's get you to agree to sit down. If you could actually collaborate in research, you could collaborate in other pandemic prevention response, because we always say that the response to pandemic is not something specific to a country. It is to be done in collaboration with other countries. So I think this consortium can actually provide sort of a vision on how countries in ASEAN, and I understand there's a friend from ASEAN here, could actually start moving forward and start having a more ASEAN-wide, sorry, my focus is ASEAN. We know, for example, in ASEAN, they have launched the ASEAN Center for Public Health APCHED. But we know that up to now, they're still struggling to have it put in place. There has been discussion that within ASEAN is going to be vaccine self-sufficiency, launched about 5-6 years ago during the leaders meeting. But again, there's still a challenge of getting that moving forward. So we see this as an opportunity to hopefully smoothen or show how it can be done. Now, secondly, as a development bank, so we're being asked, so how can we come in? So when the pandemic happened, we provided a facility called the Asia Pacific Vaccine Facility. And basically, we come in two sides, both supply and demand. So if the products are there, so what when we came into countries, most of our loans and grants, grants for some countries, were essentially to help countries buy the vaccines. And they came to us because they said that the global mechanism was just not responding fast enough. So if this consortium happened and the production is actually there, that's something we could again do, where we could actually try to provide financing that can help, in a sense, countries buy the malaria, the dengue vaccines that can be manufactured out of this consortia. We tried to provide support to countries to also build manufacturing capacity, a little more difficult, although we just had our first loan ever to Bangladesh for them to develop a government-run vaccine manufacturing plant. We have discussion with Biopharma to support them as well. And so there's also another possibility from the manufacturing side that we could explore support, whether it's government or even the private sector, because we do lend to the private sector. Now, I probably end with my challenges as I listen to the consortia. People thinking, how can we, ADB, provide support to the research part? Fortunately, we don't really have a good fit on the modalities and the financing modalities that we have. I think this is going to be my assignment when I get back to Manila, to try to see is there a way, would countries be willing to borrow money from us, for example, to create an Asian-wide NIH? Because at the end, we cannot leave the funding for Mahidol, for Chula, probably for Singapore, they may have the money, to the Gates or to GIT, and I love GIT, right? But that may not be enough. We also have to recognize that the Americans get their money from NIH, the Europeans from government. So maybe an ASEAN-wide fund to fund R&D can be created with ADB funding, providing some of the funding support with initially, something we could talk about and explore. I'll stop there.
Great, thank you very much. Steve, I'll turn to you. Again, what do you see the relationship between these consortia and the ecosystem, and what's your vision going forward?
So first of all, there's been a lot of talk about bringing regions together, and I think that certainly makes sense with the increased economies of scale compared to individual countries. I think procurement was talked about, manufacturing, there's been a lot of discussion on that, and there has been discussion on R&D, and I think Eduardo makes a good point. R&D, regional R&D may be something that may make sense. But I would also have to stress that, as we've kind of alluded to and touched on in some of the conversations today, R&D is not easy necessarily, and it's not completely predictable. And the targets are not necessarily easy. Some of these targets have been attempted for years and years and years, and mRNA is not just the magic bullet that's going to all of a sudden make it work. I think that when you're talking about this kind of R&D, you have to be talking about long term. So you have to be in it for the long term, committed for the long term, and not expecting some product all of a sudden to be available, in 3 years from now. Honestly, it's something I think when you're talking specifically about R&D that you have to think about long-term commitment. And quite frankly, the business case for any even easy RNA targets isn't necessarily there right now for any developer or manufacturer. There's things that need to evolve and develop over time, such as the cost of raw materials, the cost of ionizable lipids and capping. Huge roadblocks to make vaccines competitive. Those will eventually evolve and become more competitive. But I just want to stress the point that if we're going to do this, it's got to be with the long-term vision in mind and with knowledge that things will not be the same now compared to 5 years from now in a lot of different ways within the ecosystem.
Thanks very much indeed. A little bit of reality check there. And finally, Gabriel, what's your vision for the future? You're on now.
Hello. Thank you so much, Charles. Let me just say that one, scientifically very exciting new platform, which like few others allows fairly easy plug-and-play versatility. So extremely exciting scientifically. Two, any ecosystem, which is your question, that is based and grounded in LMICs, like this one is a global public good for equity per se. And I think it's really not too much to belabor this point because, as we all know, the most recent example of vaccine inequity suffered by LMICs was the failure of COVAX. To this day, if you actually look at some of the COVID vaccines, which are no longer in demand, but even when there is demand, what is supplied to most of the low-income countries are still the bivalent variety, refusing to recognize immune imprinting as a true scientific concept. I just came from Geneva, and I told both WHO and Gavi of this, even though Gavi is still pushing bivalent Omicron vaccines out. And of course, in this region, the PIP framework, originally because of the Indonesian government's reaction to avian flu sample sharing, back when Margaret Chan was still WHO DG, is now facing the same sort of hurdles and challenges when you're looking at the draft 1, draft 2 of the Pandemic Accord, which is 20 years after IHR revision in 2005. So I think we should not belittle the importance of an LMIC-based ecosystem. And for that, I'm very, very grateful and privileged to be in this gathering. But the third thing is, as Charles, you put it, a bit of reality check. The mantra in really vaccine development is you are either the first in class or the best in class. For all the vaccines that we've talked about, we're not the first in class. So therefore, we have to be the best in class. And by best, especially when you're talking about access and vaccine equity, you have to consider not just the effectiveness, but the cost. And on the effectiveness bit, we heard a little bit about in the most advanced of these, which is the dengue platform that Jerome was summarizing so beautifully for us. There are still fairly important challenges in terms of correlates of protection. And we have to go really deep into the science. We can't do the hand-wavy, oh, this may be CMI. Which bits of CMI are you looking at correlates of protection? And even if it is bits of CMI, it's usually very, very expensive to run standardized CMI assays that you could use, say, for the WHO PQ. And nobody has really figured it out yet. As I said, I was in Geneva for a whole week last week, and the correlates of protection agenda actually made it for the first time for 2 other bugs on SAGE's agenda. One is fairly advanced, group B strep, and the other not so quite advanced, chikungunya, which is actually a disease endemic in this part of the world, for example. So I think that that's not something that we should take as a trivial concern. And I think that goes to the feasibility, because if you can't rely on good correlates of protection, then you need to run very large and therefore very expensive efficacy trials. And that makes access difficult. And these are some of the things that we should be thinking while we're even in phase 1 of the desktop review. Then the second bit is the cost, which ultimately determines access. An mRNA platform is not usually known as the cheapest platform around. Not only in terms of manufacturing, which with scale you could push the price down, but in terms of storage, in terms of distribution, and in terms of administration. And I think that really brings us full circle back to that access issue. And by the way, access is usually actually based not on R&D, where you do the R&D. It's actually where you have the manufacturing capacity. And that really determines whether a region or a group of countries in, say, this part of the world actually gets access. And that's a very vivid, acute lesson from COVAX. And finally, let me say that I know that we're, as a panel, standing between you and dinner, and most of you have been sort of glancing sideways at the exit signs, which should also apply to what we do here. especially when you were quite early in the process. So what are the contingencies and what are the plans B, C, and D in case things don't go linearly, and things generally don't? And so how can we quickly repurpose, go back into those circles, and then really get on with the progress according to those Gantt charts? Thanks very much.
Thank you very much indeed, Gabriel. Okay, so we now have a bit of time for Q&A. If you'd be very kind to those in the room to say once again who you are and where you're from, because there are lots of people online. And if you could say who you're addressing your question to, whether it's the whole panel or individuals, and that applies also to people asking questions online. Okay, floor is open. Hello.
Alan Boekenoog, I'm with Hillman Labs, leading R&D. I want to ask the obvious question because you're on the side of potential donors, except for Petro maybe, but for the potential donors, I would want to ask, what would make it more attractive to you for us to come forward with proposals? What do you want to see in a proposal to be better convinced of supporting this?
I'll go ahead and start. From our standpoint, obviously we cannot address the whole ecosystem of mRNA. There are pieces of it that I think make very good sense to us. mRNA, in terms of an outbreak, epidemic, pandemic preparedness, is maybe the only rapid response technology platform. So we are very interested in certain targets within our programs, priority pathogens, but we're also interested in moving forward the technology that allows improvement or validation, quote unquote, of the platform. So things that address the moving forward and progress of those specific priority pathogens or the technologies that will allow rapid response in the future to an unknown pathogen. So within that framework, then how does it fit? At least for CEPI, this is a general approach.
Thank you for the question. From the Wright Foundation's perspective, I think I would like to echo the words that Jerome used, starting with the end in mind. So at least from the Wright Foundation, we would really appreciate clear articulation of your end-to-end vision. And I would respectfully disagree with maybe one comment raised by Gabrielle. I do believe that it's extremely important to ensure access from the start of your R&D. Your efforts to optimize the thermostability, the ease of operation, and the regimens that you are envisioning for scalability, all those considerations we would like to see clearly in the proposal. And also two concepts that Martin this morning briefly alluded to, I would strongly agree with. The concept of probability of technical and regulatory success, but also importantly, probability of policy development and procurement. I think a number of you, I think maybe including Eduardo, have mentioned the importance of thinking about your game plan to take the funded product beyond R&D and to have a clear path to public procurement so that the funded products are actually made available within the local public health system to ensure equitable access. So from Wright Foundation's perspective, articulating that end-to-end vision from the start, that would be important.
I think the first one I would like to say, I already said it, language of regional cooperation, highlight it. It's a priority of ADB, so it's something we would like to fund. Second, the link to pandemic preparedness and response, it's not coming out. We know there's lots of, the pandemic fund is in the World Bank right now, but there's still a lot of appetite for development banks, for even other donors. If you could say this is a way for countries to prepare for the next pandemic. So it's not in the discussion. And of course, you frame a bit that way. If you build the mRNA factories like in biopharma, you're not just preparing for malaria or dengue, but also for the next COVID-19. And last one, if you listen to the climate change and health discussion, I disagree with this because I believe that any strengthening of health system is climate adaptation. But climate change, the traditional ones, we usually say that when you talk about climate change and health, it's malaria and dengue. So if you start framing this as a climate change adaptation, you might get some climate financing as well. Because in a sense, if the climate change people only think it's malaria and dengue, they might as well get their money. But nonetheless, so these are different. So you start framing this beyond an R&D story, an R&D consortia story, but really something that's responding to a lot of global health priorities right now.
My turn. Well, first of all, let me just clarify that while I absolutely agree R&D is where we start, but let me reiterate, not having proper equity considerations in the R&D is a non-starter. But having it doesn't mean that you're going to get access, as we've learned in COVAX. Now, what we would very much like to see is more emphasis on the sustainability and the robustness of this ecosystem, because I think that is worth the most in this gathering. Whether it's any of these 4 bugs or potential vaccines or none of them is in the long term immaterial. What is most valuable is that there is this ecosystem that will always be in place with contingencies and plans B, C, D, and E, such that we would have this in place for this part of the world for equity of access. So to me, that is the most valuable part. That's where the investment, the sunk costs, will never be lost. The second thing is, of course, just really on how do we get this best in class? Do we have all of the components that I outlined? And I think that's very contextually and pathogen specific. And that, I think, is important, but probably secondary to this ecosystem that you're all building together.
I just wanted to mention something about having the end in mind. And I think that that is true, that having the end in mind is important. But I think having the end in mind with the presumption that the ecosystem exists to get to that end right now is not necessarily true. And it is different for different target pathogens. If there were huge commercial value to some of these targets, then huge commercial entities are going to be pursuing them. So I think that we have to think exactly, this is something that I think we've gone through an evolution in at CEPI, is that we're trying to do things, but there may not necessarily, let's say, be the motivation to do it all the way, end to end. So sometimes it's hard to have that line of sight. So we actually are pushing the envelope. We're pushing this discussion, and I think that's the main thing that we have to do, is indicate where are the gaps in the ecosystem that need to be filled, not necessarily by an individual organization, but by the whole global community. And I think there's a lot of those, especially when we're talking about pandemic preparedness, and especially when we're talking about pandemic preparedness in the context of equitable access.
Hello, my name is Hun Sang Lee from the Right Foundations. Going back to what had been just said by Ingyu from the CEPI, what would be the gaps to fill? I just was wondering about what had been mentioned this morning about the importance of the regulatory bodies. My question, especially to Eduardo and any of the person in the panel, is there had been a report from ADB, lab to jab. So really it talked about the whole ecosystem of how to go from the research laboratory to all the vaccinations to the end users. But in the process, I have noticed ADB had been having quite a bit of effort for the regulatory harmonizations in Asia Pacific and regulatory capacity building. And there is some of the renowned regulatory body in this part of the world. We have Japan and Australia, 2 SRAs, and now we have Korea MFDS and Singapore, who has been listed as WHO listed authorities. And there are additional efforts for strengthening regulatory capacity throughout the region. So what do you think that efforts will take place in this formation of the regional R&D consortia effort? Thank you.
So essentially when the pandemic started, so when we started mobilizing the AFVAX facilities, 9 billion, so the decision was the vaccines has to be either WHO PQ or stringent regulatory authority. That was to mitigate the risk that you get the vaccine doesn't work. Of course, the problem was you end up only buying vaccines that came from those countries. And when we started having vaccines from other countries, which were not stringent regulatory authority or not WHO PQ, so for example, from China, or even vaccines that were contract manufactured in other countries, then we could not pay for that. So we started to say, how do we resolve this? We still need to manage the risk. So we started convening the different regulatory authorities, and with the end objective that can we find a way in which we could, in a sense, get to acknowledge, for example, BPOM in Indonesia, and try to accept that when it signs off by BPOM, then it was okay. But the pandemic ended before we find an answer. But nonetheless, what we have done now is we started to provide support. For example, in Indonesia, we are been providing to strengthen. So Southeast Asia is lucky, and Asia is lucky. In Southeast Asia, we have BPOM is ML3, Vietnam, Thailand, Singapore, Indonesia. So we have enough regulatory agency here that will be comfortable. That if there's when these vaccines comes out, we could just basically say if it's manufactured in Thailand or biopharma, it's fine. South Asia a little bit more problematic. It's only India that's good enough. So we decided to provide some support to Bangladesh now. Central Asia. So in a sense, that's our approach right now. We always dream of that all of the regular agencies would be harmonized, working together. But I think it's a little bit much more. We probably need to do a little bit more country-specific approach.
There's actually a question online for you, gentlemen, which is, and this will be the last question because we're running out of time. I don't know whether you saw it, but what it is most of the dengue vaccines licensed and under trials are live attenuated, making safety a key challenge and concern in the healthy and pediatric population. Do you think the novel platforms such as mRNA will solve the concerns of safety and correlates of protection?
Not so sure how that question come to me because it's dengue, right?
Oh, sorry, it's dengue. Yes, sorry, I read it out without thinking. I just assumed that the question online. Sorry, Jerome, it's going to go to you. Sorry, gentlemen, I just read it as it was without thinking. I apologize. I'll read it to you again. So most of the dengue vaccines licensed and under trials are live attenuated, making safety a key challenge and concern in the healthy and pediatric populations. Do you think the novel platforms like mRNA will solve the concerns of safety and correlates of protection?
I don't think they're necessarily going to solve the question around correlates of protection unless we design the trials to answer them. I think that around safety, again, there are still concerns in some groups about the use of mRNA vaccines in children, to the extent that pediatricians have told me that they would not have their children vaccinated with mRNA. So I think we have to expand databases around all vaccines, particularly around safety. And the fact that they're mRNA, I think that the key considerations for vaccines for dengue, the generation of sustained cellular and humoral antibody and humoral responses that are directed against the virus antigens that we believe are important for control. And if it's an mRNA, if an mRNA vaccine will do that, that's great. You're right, the current examples are replicating virus.
Thanks very much indeed.
science.
Sorry to put you on the spot like that, and particularly sorry to Jetson, dragging her up here for nothing. Sorry. So just before I invite Duce Medini to come and give some closing remarks, I'd just like to give a really big hand of applause for our panel. Duce.
Thanks, Charles. It's a great pleasure on behalf of the Scientific Advisory Committee of the RNA program. First of all, to congratulate MPP, the WHO, the research consortia that have been formed for the great achievement of the signature that happened today. And then, since I'm aware that I'm really the last one standing between you and the dinner reception that is waiting outside, I will share only one consideration with the group, which actually builds on a theme that has been raised by many, particularly comments from Gabriel, Inq, Robin this morning. In order to get to the financial sustainability that Martin introduced opening this colloquium, I think we are facing a conundrum that I will characterize as an impedance matching problem. We have product development on one side, we have manufacturing technology, development validation on the other side, and these two things may not go at the same speed. And that's the point that I would like to attract your attention before leaving you for dinner. Everyone that ever developed manufacturing technology knows that there is no manufacturing technology without a product. Only when you get in front of regulatory authority with your GMP process, because you're submitting for clinical use of your technology on a certain product, it's only at that point that you understand what you actually have in your hands. Is my process good enough? Is it worth, etc. Now, the research products that we have discussed today here, not all of them are potentially ready to provide manufacturing technologies with something to be tested in humans over the next 6 to 12 months, which is actually the timeline for some of the manufacturing technology that we are discussing. Now, this is where we have an impedance matching problem. Some of what we have discussed today may need 3-5 years, if ever RNA is actually the right technology to address that problem. So what do? Do we wait 5 years before we test our manufacturing technology? Now, this is what I would like to propose. This is not just a timing issue. It's also a problem of financial sustainability in terms of volumes. Not all the products that we have discussed today, if successfully developed to market, would have a volume to market that would justify the financial sustainability of a fully dedicated manufacturing plant for that product only. Some of them may. I'm thinking of a dengue vaccine that would definitely have a market, if ever to market, that would justify an entire manufacturing line, but maybe not necessarily all of them. So here is where we should remind ourselves that we are not talking any manufacturing technology. We're talking RNA that comes with an incredible platform potential if used in a multi-product standardized way. Here is where we need multiple products to test synchronously at the same time as much as possible manufacturing technologies that can demonstrate in this way that the same line, without technical adaptation, without tech transfer, without revalidation, can be used on more than one product. Now, in order to achieve this, I think that the regional consortia that we are celebrating today, launching, are ideally positioned to support this interaction between manufacturing and product development if they remain open to the global coordination support provided by MPP and WHO, particularly in order to favor technology standardization. What I'm trying to say is that if every product tries to focus on one manufacturing solution, this is going to be hardly financially sustainable at any time, neither now nor at the end of the process development. So what we need is standardization and access to the same manufacturing platforms across the different consortia. And if we do that, we have a concrete, realistic shot to financial sustainability in terms of timelines and volumes, which will achieve 3 major objectives that are so close to the hearts of everyone in this room. First of all, equitable access to the products that get developed, which is unachievable without financial sustainability. Second, the return on the investment that is generated by financially sustainable manufacturing can re-inject resources into the local, regional R&D ecosystems. And third, we would have as a byproduct for free pandemic preparedness. Because if a manufacturing technology is truly demonstrated to be multi-product, by manufacturing more than one financially sustainable, non-pandemic product, then it can also manufacture a pandemic response product. And that's how we get it for free. And with this, I think I will leave you to the reception. Thank everyone for the great day. And I think we give the appointment to the session tomorrow morning. Is that right? Thanks, everyone.